everyone, and welcome to Biology Buddies. In today's video, we'll be discussing the Hershey and Chase experiment. In 1952, two scientists, Alfred Hershey and Martha Chase, conducted an experiment to prove that DNA is the genetic material. There were two different groups, one to test whether protein was the genetic material, while the other tested whether DNA was the genetic material. T2 bacteriophage, the viruses were grown in one of two isotopic mediums. One was grown in a medium containing radioactive sulfur 35S for radio-labeled proteins. Sulfur was an element found in proteins due to its disulfide bridges, but not in DNA. The other was grown in a medium containing radioactive phosphorus, 32P, for radio-labeled DNA. Phosphorus is present in DNA due to its phosphate group, but not in proteins. The viruses on those two mediums were then allowed to infect Bacterium E. coli after they blended the mixture to detach viruses from the bacterial cells. A centrifuge was used to separate them, bacterial cells from a solid pallet at the bottom of the tube, while viruses above in the liquid supernatant. Afterwards, it was observed that 35S, which means the protein, remained in the supernatant, while 32P, which means the DNA, was found in the pallet where bacteria was. This proves that DNA was the genetic material of the viruses, since DNA, which was radio-labeled by radioactive phosphorus, was being transferred into the bacteria. Thank you so much for watching our video on the Hershey and Chase experiment. We hope it helped you to understand this experiment much better. See you again next time on Biology Buddies.